this lab doesn't do anything but rip stuff up. It's crazy out here, man. This lab doesn't do anything but rip stuff up. It's crazy out here, man. Good evening, Pittsburgh and the universe. This is JC on a bike. My name is JC. This is Journal Club on a bike. I got a ripping paper for you tonight from the Tonegawa lab, and I think as soon as I mention the name Tonegawa, you know it's a ripping paper. Because this lab doesn't do anything but rip stuff up. This is a 8-figure neuron paper that builds upon 2 papers that came out in the last couple years with the first author named Kim, also from the Tonegawa lab, where they detailed 2 subpopulations in the basolateral amygdala that extend from anterior to posterior and play a role in aversive and reward learning, valence learning. And the history of the amygdala is kind of an ugly one because it has for a long time been the seat of aversive learning and fear conditioning. And for a long time people have tried to assert that it wouldn't be very likely to just have a fear center, that it may be more likely that it would be a sort of valence circuit. But because fear conditioning is such an amazing, high-speed, powerful learning paradigm where the behavioral results of training are so striking and powerful that a lot of our understanding of how basic learning occurs is from fear conditioning, from context fear conditioning. And so in this paper, they are using context fear conditioning as a paradigm, but then they're going to look specifically at the animal's learning process when it learns to forget to be afraid. So if I explain that really quickly, what that means is that we're going to context fear condition an animal. You can look in figure 1A. Context fear conditioning is where you pair a tone with a foot shock in a particular room that's not their home cage. And so after 3 training sessions, you see there in figure B or C, I don't know which one it is, you see that after the 3rd session, they're all freezing when they hear the tone. And if you want context fear conditioning, then it's specific for the room that they're in. If you played that tone in a different room, they wouldn't freeze. So they learn to be afraid in the room, and they learn to be afraid when they hear the tone because they know the foot shock is coming. That's day 1. And then in day 2, what they do, then on day 2, what they do is they look at how the animal then forgets to be afraid by exposing them to the conditioned stimulus, that would be the shock box and the tone, but no longer giving them a foot shock. And then on day 3, we're going to put them in the context again and just see how much they freeze. Now, I have to admit, I'm not 100% sure that they're using a tone. They could just be using context, but I guess they're using a tone. I don't know. You can look that up. It doesn't really matter, I don't think, for the results. It's important for understanding how they're training the animal. But let's assume for now that they pair a tone with a foot shock. That's the conditioned and unconditioned stimuli that they're using for this training. And so what they're going to do then is they're going to record from these 2 subpopulations they've identified and see what they do during fear conditioning or during learning. And in this case, they're really interested in extinction learning. And extinction learning, again, what cells are activated when the fear memory is being erased, basically. And then what they're going to do is they're going to look at the percentage of CFOS cells that are labeled as either respondin 2 positive cells or this phosphatase something something. It's PPP1RB1. It's a phosphatase inhibitory number 1B or something like that. Anyway, cells that express that subunit are different than cells that express this respondin 2. And these are the two populations. So what they're going to look at is these two populations and see how they respond with FOS signaling in these three groups. And the three groups, remember, are the groups that just plain old got fear conditioned and didn't get extincted. And then an animal that didn't get shocked, and then an animal that got shocked and then went through extinction training on day 2. And so we're going to try and figure out what percentage of cells or who's expressing FOS during these time periods. And what they see is that during extinction learning and extinction retrieval, the PPP1R1Bs, those cells are activated, and the other population, the respondin 2 population, is actually suppressed, and they're activated during the shock. That's just figure 1. So they can see a difference in the populations, and they can follow these populations and their FOS activation as a result of these 3 different types of training. The 2 control groups and then the extinction learning group. So figure 2 is going to report data from calcium signals from these same cells to try and show, what percentage is activated, what percentage is deactivated, and what percentage doesn't really respond to these different trainings. And here again, what they show, try and hit this break here. And in figure 2 now we're looking specifically at cells that were activated or not activated. And if we look at cells that are upregulated, those tend to be PPP cells, and those cells that are suppressed tend to be respondin 2 cells. And so that's pretty cool, because now the calcium signaling is sorting out on a cell by cell basis, and of course that suggests that not all cells are involved, right? Not all cells can be involved. There should be an engram of cells. And that engram of cells then is the subset of cells that gets activated by this learning. Not all the cells. Can't possibly be all the cells. And that's what you see in figure 1 too. It's not all the cells, but nonetheless, the propensity or the number of cells that gets activated in those different things, it's really the PPP cells that seem to be activated by extinction training. And the calcium signals in figure 2 also seem to jibe with that. If you look at M and N in particular, you can see that the cells that were activated during the extinction training are somehow, active. I don't exactly get it, but they do show that the activated cells, the PPP cells are activated and the other ones are suppressed during the extinction train. And that's, in the calcium signal, they can see that too. So figure 3 is the monster figure. Figure 3 is the one where you're just like, damn, they did it. It's up and down. And up and down figures are pretty tough to find in neuroscience, but this is an up and down figure. So they're going to manipulate both populations of neurons up and down. The PPP neurons are going to be manipulated up and down, and the respondin 2 neurons are going to be manipulated up and down. And correspondingly, you will see that when they manipulate the PPP cells up, then extinction actually occurs earlier. I think that's an E. And then they also show an H that they can, no, that's a different figure. Sorry, I got to think again. This one is up and down, up and down. So when they manipulate the respondent 2 cells, then they can eliminate extinction learning when they activate them. That's right. And when they inhibit them, extinction learning is normal. And when they stimulate the PPP side, I think they get a quicker extinction, and when they inhibit it, then they don't get extinction. And they can see that because the animal freezes, and that's pretty badass. So if they extinct, that's during extinction training, and so they're going to turn the light on during extinction training. That's what the cool part about figure 3 is, if I remember it right. Shit. It's 8 figures in Neuron, man. Give me a break. So figure 5 is then going to bring in the Tonegawa magic and use doxycycline rather than labeling all the cells that express one of these 2 markers. Now they're going to use doxycycline techniques to label the specific engram cells that are activated during extinction training. And what they're going to try and do by capturing those cells is just activate them with the light and see if they can induce extinction without the actual behavioral paradigm training. And stupid, badass, of course it works. Optical extinction works when they hijack these particular neurons. And that's pretty amazing from the perspective of understanding how the brain works. That child needs to get out of my way. And so, if they hijack the engram of the PPP cells and put it under the control of channelrhodopsin, they can use those engram cells to induce extinction with the light. Super badass. Now, figure 6 is kind of a detailed figure that I'll just give you the brief overview of, and that is that they show here that there's a lot of overlap between cells activated by reward and those cells activated by extinction learning. And so they're starting to hint at the idea that extinction learning and reward learning might just be the same thing to these cells. Activating these cells is positive and activating the other cells is negative, and they are interacting populations. So the only question is whether the first population can suppress the second one and vice versa, or if it's really a one-way street like this paper tends to indicate. And so figure 7, what they're going to do is fool around with the reward engrams and the extinction engrams and show, I believe they show that they can use reward cells for extinction to drive extinction, and they use extinction cells to drive reward. And it's overall just showing you how awesome and complete their results can be. Oh yeah, so figure 7 shows, I think, not just that they are the same, but their calcium signals are similar. Calcium signals between reward and extinction are similar. And then in figure 8, they hijack those cells and stimulate them and show that by stimulating that population you can get evoke reward type behavior or evoke extinction type behavior. It's the same population of cells, and it doesn't really matter how you recruit the engram. Those cells are capable of doing it. So the Tonegawa lab has an untold number of extremely good papers in the last 5 years or 10 years. Susumu Tonegawa got a Nobel Prize for immunology a while back, and I would argue that the one problem with him getting another Nobel is that so much of what he's done is methodologically completely inaccessible to all other labs. He's not making tools that you can just send around and everybody can use them. It's not that I don't believe these tools work. It's just that unlike optogenetics, it's pretty tough to find lots of other labs executing experiments like the Tonegawa lab. So their expertise is not as easy to transfer as you might think. Yeah, Steven Ramirez is the lab, but you know, he trained in those techniques for 6 years or something. So, you know, in 6 years people can learn to patch clamp too, but that doesn't mean that patch clamp is very accessible to somebody who just wants to get out a how-to book and learn how to do it. And with optogenetics, you don't need a how-to book, right? You just inject and fire. But the genetic tools that are used in this paper and leveraged regularly in the Tonegawa paper are, to a large extent, beyond the reach of normal labs. The money, the infrastructure, the expertise, it's hard to assemble. And so this lab is going to continue to dominate this in vivo field of capturing engrams, simply because very, very few other labs can do it. But man, it's a cool paper. I mean, it's the polar opposite of recording from the entire cortex and explaining it with correlative measures and linear models. It's exactly the opposite. So thanks for joining me on my ride. That was the latest paper from the Tonegawa lab at MIT. As usual, they destroyed another open question in science. This time, over a period of about 4 years, they've identified a couple different populations in the basolateral amygdala that extend from rostral to caudal. They show that these two populations tend to be up and down populations in the sense of one is more activated by the shock and the other one is more activated during extinction training. And they go on to show that extinction training requires the activation of one population and not the other, and that this population might be suppressing the first. They also go on to show that the second population, the population they're interested in, this PPP1R1B cells, are cells which are also encoding for reward, and that their FOS signal, the same number of cells are labeled by FOS when we do reward or when we do extinction learning. And so they use calcium signaling, they use FOS counting, they use arch and channel rhodopsin activation at particular times during training. It's simply one of these juggernaut papers that this lab has become known for, where you get causality and you get effect and you get all of this stuff just packaged into one paper. And I would suggest that it's only in Neuron because it's just got too much stuff for Nature. Nature can't publish an 8-paper figure anymore. And Neuron is the only real high-level journal that can take a paper like this and expound on it in the way that they have. It's an excellent paper. It's entirely worth reading, as are the two papers from Kim, the first author from the Tonegawa paper earlier, 2015 and 2016, where they detail these two populations and identify them. If you liked it, like and subscribe, and I'll see you next time on JC on a Bite.